Neuroscience and beyond. Know more, get inspired. Welcome to the Neuroscience and Beyond podcast. My name is Vilen Georgiev, and I'm a PhD student at the University Medical Center Göttingen in Germany. Together with my PhD colleagues Sabina, Christina, and Lenny, we desire to bring science, and especially neuroscience, closer to the general public. In the following episodes, we're going to talk to various scientists about their inspiration to do science, their research projects, and their opinion on current scientific problems. Today, our guest is Professor André Fischer. Professor Fischer is the head of the German Center for Neurodegenerative Diseases in Göttingen, Germany. His research focuses on the mechanisms that underlie Alzheimer's disease, schizophrenia, and anxiety disorder. In addition, his team studies the impact of protective environmental factors such as physical and cognitive exercise on the onset and progression of Alzheimer's disease. One of the main goals of Professor Fischer's lab is to identify epigenetic drug targets and to establish epigenome changes as suitable biomarkers of disease progression and therapy. Before we begin, I would like to mention that this podcast is separate from my research position at the University Medical Center Göttingen in Germany. This podcast is supported by the International Max Planck Research School for Neurosciences, as well as the Cluster of Excellence Multiscale Bioimaging in Göttingen, Germany. And now, please enjoy the talk with Professor André Fischer. Hello, Professor Fischer.
Hello.
Thank you for being here. You have a successful scientific career. We are going to discuss about your projects, your work a little bit later. But before we start with that, I would like to know what inspired you in first place to get into science.
Well, actually, I can tell you what got me into science because that fits very much to what we have here. So we're in the German Primate Center right now in a beautiful seminar room. And when I was still going to school, I was super fascinated by the work of Richard Leakey, basically, and Jane Goodall, for example. So I wanted to go to Africa and dig for some bones of our ancestors or study monkeys and learn something about where we come from. And then when reading these books, I was super fascinated. Going then into reality and see what this really means, basically, that was a little bit disappointing. And I figured out that that's not what I want to do. Because I went to some lectures of people who were doing this kind of research, basically, in real life nowadays. And that was nothing like Jane Goodall or Richard Leakey or these heroes of my childhood. That was actually pretty boring work, as scientists or science sometimes is. And then I decided I want to do something more in the medical area and also work on the molecular mechanisms. So pretty similar, actually, trying to understand how life works. And this was the driving force. Molecular biology was really something I was intrigued by, and gene expression already back then. And that it became the neuroscience was more of a coincidence because back then when I started to study, I got exposed to neuroscientific questions. And of course, one of the most fascinating questions in my view is still how the brain works. We still don't understand. It's debatable if we will ever be able to understand it with our own brain, to understand how the brain works. But at least I think we can understand what goes wrong when certain things do not work anymore. And this is how I became interested then in neurodegenerative and neuropsychiatric diseases, and this is where we still are today.
That is great. So you studied in Germany, and afterwards you had several years in the US, correct? And as I remember, you were there in top-notch institutions such as Harvard, MIT. Could you please make a comparison between the way of working there in the US and in Germany, for example? Is there any difference in terms of pace, in terms of stress or facilities?
Yeah, there's a difference, of course, cultural-wise, basically, but also in terms of research. And I cannot compare this, of course, to every place in the US. But as you mentioned, so let's compare Germany to the elite universities, such as Harvard, where I was, and MIT. And even Göttingen, actually, where we are nowadays, considering itself a kind of elite university. And most of the people have a totally different drive. And this is the complete attitude because there are just much more people and they're really working hard. And I give you an example. If you go to Harvard, actually, in any kind of research lab, Sunday, midnight, it's going to be full of people working there, hardcore. So they also, I mean, some people come late. I mean, it's people from all over the world, but they do have this drive that they know they have 3-4 years, and if they want to be successful, they have to work hard. There's of course much more competition, and that is not guaranteeing success, right? But this actually, when I came back to Germany, and also, well, it was different actually. I think when we were around, but that was also because it was a small group and things like this. So this, for the large part, is very, very different. Which I'm not saying it's good or bad, but actually this debate that we have about work-life balance is certainly a little bit more on the life part, which is totally okay. But of course, this is why actually I think people who make it then there also will be successful, for example, because these are the 3 years in your life where you have to really work hard, and then later on you can be a little bit more relaxed. So this is one thing that is different. The other thing that is also different is that it's much more driven by money funding. So, for example, in Germany, if I compare it, you have Max Planck institutes, you have Helmholtz institutes, and they are super well funded still with core funding and board funding. So people don't have to apply for third-party funding, which in the US, even at the big institutions, if you don't have money from the grants, then it's difficult to survive, specifically for the younger people. And you're also exposed to this by a student because you are always involved in writing R1 grants and things like this. And there, of course, they don't have time, let's say here, so to do research for a longer period without publishing anything. Something that you can do in Germany, actually, developing new techniques that might take you 5 years without any publication, that would be much more difficult in the US. Yeah. On the other hand, the US, in my view, if they really want to push something forward, there's this saying actually, go big or go home. And that's what they do. So I can tell you from our field, if they want to say we want to sequence 1,000 postmortem human brains and do the complete characterization of these, then there will be the money to do this if somebody whose stakeholder decides to do this. And that is very, very different normally in Germany, where there's really huge projects that have big impact then on the entire field. Usually you don't have this because it's more smaller funding schemes and things like this. So these are, I think, some differences that come to mind right now. And again, I'd like to reiterate that this is not any kind of evaluation. So both has advantages and disadvantages, because when we were still at the INI, we were once discussing something like, for example, Erwin Neher, our famous Nobel laureate here, developing the patch clamp technology, which in the end was a lot of technical things and playing around. There might be a reason why this was developed in Germany and not in the US, for example.
You mentioned the drive of the students. Can you elaborate? Do you think this drive comes within the students, or they are pushed by their PIs, or is some kind of mutual activity that, okay, your PI kind of pushes you and I need to work a little bit more in order to achieve this? Because Harvard, MIT, these are extremely international institutions, right? But also Göttingen, we have here, at least in our graduate school, people from all around the world, right? And for me, this should not be cultural. There should be something else. Well,
it's just this atmosphere. Of course, it's also much, much bigger than here. I mean, Göttingen is still a comparatively small city, although we have a lot of students. But it's both. So if the whole atmosphere is there, basically, that for the time you're being there, I mean, your working place is more or less your home, then everybody's doing this, right? And you don't need to push people to do that. And I think it's also fun. I think there might be also some differences, because the institutions there, they do a lot for their employees. So let's take a few things. Health insurances. In the US, I think we all know that this is a complicated issue. But if you're enrolled with one of these big universities, you have full access to their health insurance plans. So they take good care of you. And so the other thing is also that you don't have to go anywhere else to do what you like to do. So, the research institutes, they are huge. You take the elevator down to the first floor, there's a gym, there's a pool. So you can, during your break, you just do your workout, which in Göttingen is also pretty good, because you just have to go here to our sport institute, which is not far away from where we work. right? But then you also have the restaurants and cinemas. Everything is nearby. So I think there's a strategy that they, have all the things that you can just do in between. For example, let's say you do an experiment, you have an incubation time for 2 hours. So now you can read a paper, or you go to the gym, or you go to some kind of restaurants and things like this. So it's a different atmosphere. And then everybody's doing like this. So if you don't do it, actually, then you're kind of like a little bit weird, right?
Yeah, you're not effective, right? You cannot achieve these goals that you...
And the other thing is, of course, that there's a very different spirit in terms of getting donations from alumni, but also from other rich people that, have a relative or themselves suffer from this or that disease. And then there's this willingness actually to really donate just money for research. So, for example, a lot of people that I know over there, they do run on grants, but they also run on money from just people who put 50 million on the table because her spouse has Alzheimer's disease and things like this. And I always wonder, why is this the case? Because we don't really have this here. Of course, there is also this competition. So if there's a new research building, like we have now the Heart and Brain Center in the US, there would be actually a call, do you want to have your name on it, and then you have to pay something, and then the building is named after you. But that, of course, brings in good money that you can use for anything that you want. And the other thing that I always think is actually, so to study in the US is, of course, super expensive. And here it's still almost for free, right? And maybe the human brain works like this. If it's for free, it's not good enough. I'm not saying that's the case. But this is why you are maybe more inclined to give something back, because you really had to suffer to obtain something like this. I don't know if this is true, but I still remember when I came to Göttingen, coming back from the US to the European Neuroscience Institute. And we were doing back then, from the cluster that we had, this CNMPB, some kind of like talks to the lay audience in a bar downtown in Göttingen here called the IMPRS. And then I had this idea that maybe we just ask people actually if they want to donate something for research. And even if they only if in the end you have 500 euros, you could buy an antibody, which for the young groupies was good. And then I had a very interesting debate with some of the people of the board of directors of the DZNE who were, of course, completely against this. But they also couldn't explain me why. But it's just not the cultural spirit to do something like this, which I think we just have to accept.
Exactly. I think also that here in Germany, people are less inclined to market their ideas, so to say, if I can express myself. And in the US, you have always this, you have to sell here. You sell your science, sell your products, etc. Maybe this is also influences exactly this aspect with the foundation. These are mostly foundations, right? So a rich family that...
Well, sometimes it's foundation, but it's also private people. But you had another question, which I may allude to. So you asked if the students are forced. And of course, this also exists. I think this exists here. I've seen this, or I heard from colleagues and in the US also that, of course, the PI wants you to work hard as a student or a postdoc, but also that they control if you're working and how long you're working. But this actually, to my experience, didn't have much of an effect, because if somebody has the drive, then this person would do it. If not, then you can also not force it to this, because what I observed, not in the labs that I was, but from colleagues, that everybody knew that the PI Sunday morning, basically after the exercise, would come to the lab at 10 o'clock and then would leave at 11. And then everybody was there from 10 to 11 and then left. So, I mean, this is, of course, making not too much sense, right? And I mean, the time that you are there should also not be the measure of excellence, but the data that is produced, right?
Exactly. Yeah.
And this, I mean, we all know that science is kind of unpredictable. You can have a hypothesis and then you do experiments and then the experiments may not work. So you have technical issues and then the hypothesis is not proven to be wrong. You have to do other things, so it takes a little bit longer. Or you're super lucky, everything works from the first time, then you just go ahead.
Unfortunately, lucky is scarce.
Most of the time, actually, it's a little bit of try and error.
Yes, exactly. People say 90% failure, 10% eventually success, then you can be happy and have your paper.
Yeah.
So you mentioned European Neuroscience Institute, which is in Göttingen. It was funded by Erwin Neher and several other scientists in Göttingen. What made you change the direction US, go back to Germany, and exactly coming to Göttingen?
That was actually, as often in life, a pure coincidence, and that was also not planned. So when I was in the US, I had a really great time and I liked it there very much. And so Boston is also a very vivid area. It's a beautiful city to live in because it's very European, a bit cold in the winter, a bit hot in the summer. But scientific, it's of course just great because all the big companies are there, all the greatest research institutions, so very collaborative atmosphere. And when I finished the postdoc research, there was Merck, the US Merck company, who just built a new research institute next to the Harvard campus, the Longwood campus for the medical school. And they were recruiting people like crazy. And so I had a job offer there. And if I may, there's a very interesting anecdote that I can tell you from interviews in the industry. So I had to go there and give a talk, and then the head of this center was not there because he was somewhere else. So they taped this thing, and then I talked to some scientists, and then the next day I had to go to his office. And then I went there, and this was really, the office was probably 4 times at this room, and in front of this there was a buffet, just all the time. And then I went to his office, and he said, your talk is very interesting, and I listened to it in double speed, because the people talk so slow. Always when I have breakfast, actually, I'm listening to the talks of these candidates, because I still understand. So it was a very interesting character, and we had a nice chat. And I also had an offer in academia. But then in the end, we had also the children born. My wife was over there, and she was originally from Iran. So she was German. But then, it's somehow still difficult because you had to have this 2-year home residency. So for private reasons, actually, it would be a bit difficult actually to have a transatlantic relationship with a small child if I would stay in the US. And then actually one night I looked actually at the internet and I saw this position there in Göttingen. And I wrote to Erwin directly. I never talked to him before. And I asked actually, so what is this about? Should I apply? And then he said, yeah, so next week we have a search committee. Why don't you come? And then I flew over in a week and came to the European Neuroscience Institute. And then I was super shocked because I didn't know how they do it in Germany. And in the US, if you go on the job market for academia, they invite every candidate individually. So you go there, you go to the university, then you are there 2 days, you talk to all the faculties, you give a talk, and it's nice. And then you go away, and then they invite the next one. This is, in a way, nice because you don't feel the pressure of competition, but also it takes some time, right? And here I was coming, and then I saw, okay, so there are many people, actually. And then I realized, okay, these are all the guys that apply for the same position. And they all we were all in the same room, and everybody was giving a talk, and you listened to this. So this I found super strange, to be honest. But then I realized the advantage is that obviously at the evening, the board of directors came together and made a decision on the same day. And then they came to me and said, do you want to come? And I said, well, let's talk to the family. And then the next day I said, yes, I want to come. And then it was super fast, I have to say. That actually, when you go to the faculty, to the university, because there's a little bit more administration associated with this, it's not that fast. But this construct actually being also not 100% university, but also Max Planck involved, and they could take decisions very, very rapidly. This was really something I liked then, and I understand why, of course, they invite then everybody at the same time, which they still do. So it's just a normal procedure for every kind of position here in Germany, right?
Mm-hmm. Yeah, I've also been, I've attended once at FMP in Berlin, mm-hmm. the same thing. They applied for a position. It was very interesting that okay, these guys now, we are fighting for one position, and exactly. We gotta make it. We gotta.
So I was a bit naive. I didn't know about this, but then of course now it's. I know it's a normal procedure. Yeah.
Exactly. And then, according to your CV, which is available online, you accepted a position at DZNE, the German Center for Neurodegenerative Diseases, but you declined another one in another institution in München.
Yes.
May I ask you what made you stay here again? Because München is a very competitive one, right?
Yes.
And it's very interesting to me personally to understand.
Well, so the situation back then was that at the ENI, back then, there was no tenure track option, right? So now it exists, which is also a good thing, actually, when the evaluation criteria are right. And back then at the ENI, there were a couple of things that were interesting. So first of all, this is also why Erwin Neher, Dieter Brichter, and Walter Stümer and all the people from the first generation came up with the ENI. That was not planned as it happened then back then, because they said there's no structured path for young people coming after a postdoc and going into academia, into professorship ranks and things like this, what we have now. And then they thought, okay, so how can we best prepare them? And the idea was to put them all in an institute, give them complete independence, and at the same time, let them do the administration, and this all by themselves. So the ENI still, I think, is kind of administered by group leaders. Of course, there's a board of director, but the day-to-day stuff you have to do yourself, including getting exposed to how you do Dienstreiseanträge and all this stuff. And if you have suffered through all of this once, actually, you know that this is really no fun. But I think it's good to do it from time to time to understand how this and other things really work. And so this was actually nice. So there was a lot of freedom, and you had 5 years to do research, plus 2 that you can then extend if you were doing well. But then you had to leave. So at some point they were saying, that's over, just leave this. And then, so yeah, if you publish well, actually then after 4 to 5 years, you go on the job market. And at this time point, there was this interesting idea from our former Minister of Science, Annette Schawan, who is actually a very impressive person, I have to say, somebody who really knows what she wants and then is fighting for this. And she said, we need something like the NIH, so we need big research centers to tackle the major health issues that we are facing right now and will face in the future. And this, what they identified was cardiovascular, cancer, lung diseases, and amongst this also neurodegenerative diseases. And then they really said, we put a lot of money on the table, this was 2008, to fund these new institutes. And it was not really clear how this would be implemented. And the Center for Neurodegenerative Diseases back then, that was... this was then later on becoming a Helmholtz Institute and not project-based funding. So the idea was that there was a call, and then every university could apply. And the structure was the following, that there should be a center institute, like a major site, and there should be satellites based on their experience. Mm-hmm. And the concept, I think, this is for all the healthcare centers, is very interesting because you pump money into something that's not entirely new, but you build on the existing structures that you have. Okay, and then Göttingen applied with others, and we were actually in the evaluation for the main headquarter. And the person who was mainly pushing this was a neurologist that is now the head of neurology in Aachen. So this was Jörg Schulz, who back then wrote the application. And then we had to go to Heidelberg to defend our application for this center. And our competitors were Bonn, Munich, and one other place, which I forgot now actually, which it was. And then I went there with Professor von Figura, I recall, and Jörg Schulz, and just 3. And they took me because I was the young guy at the ENI doing Alzheimer research and this type of things. And and Christian Griesinger was also there. So he came from skiing actually and had an accident. So he was actually having, had some injuries basically, but he still made it with a car to come there. And then we defended. In the end, Göttingen was not the main center because there were political issues that Bonn should become one of the centers, of course. But we were supposed to be a satellite. And then we were driving back and were also very happy. And I still remember that von Figura called his wife and said, I'm driving on the Autobahn with a professor from the Max Planck who just had a head trauma, but I think I will arrive. So this was an interesting experience. No, but then we had to write. So it was clear that there's a head center and the satellites were clear, but in order to get the money, you still had to write an application. Mm-hmm. And this was the time when then Jörg Schulz left, so he went to Aachen, and then there was nobody doing it, and in the end, Göttingen was kicked out of the DZNE again. Yeah. And then a lot of people tried this, and then they did something, actually, they said, okay, so why don't this young guy does it? And so the former head of psychiatry, Peter Falkay, who's a very dear friend and colleague who's now in Munich, said, okay, so you're going to write your application, do what you want, and then I shifted the whole thing to epigenetics. And we had to reapply. But then actually we were again in the competition with Berlin, with Hamburg and Essen and Göttingen for the last site to be filled. And at this time point then we again wrote an application. We went to Bonn to defend then. And then we were doing so extremely well that they had to say, okay, so Göttingen has to be the new site now, but Berlin was also there, and Berlin was supposed to be part of the DZNE. So we had 2 new sites, and in the end it took a while, and then it was solved that there were 2 new sites to the DZNE. So this was a very interesting thing. And at this time point, I thought I was also applying for other jobs. And so I had this offer in Munich and the other one in Magdeburg, which were very attractive places, to be honest. And then the former head of this search committee called me and said, we now have to make a decision. Is it going to be Berlin or Göttingen? That was not quite clear. And they said, so what is with these things there now? So you have these other things. If we now say it's going to be Göttingen, we want to be 100% sure that you're here. And then, so what can we do? And they said, okay, so you have to decline these offers, right? Which I did then. So I did it. And then actually they said, oh, but Göttingen is not going to be the site. So this is, no, but in the end, then it worked out. So then there was this things actually that Göttingen will be a new DZNE and Berlin would be a new DZNE and everything was fine. But of course that was, sometimes you just have to be at the right place at the right time. And this is, of course, you can search for these opportunities. But if, let's say, in the end that would have been kicked out, then I would now have a fallout option and would be somewhere in the basement of the psychiatry or something like this, right? Or you just go somewhere else, and that's also a possibility. But of course, for me, this was super attractive because you could build up something from really zero, right? And that was more attractive than just going to this, have one of these positions there and do your research, right?
Yeah, I can imagine.
Which, if I would know now what kind of administrative workload this is, I would think twice.
By the way, you mentioned Dienstreiseanträge, which is a German word for a document that we have to fill out when we want to travel to a conference, for example. Yeah. And it's quite tricky to make it happen. At least we had some colleagues suffering that cannot speak German. But yes, Germany is famous for the administrative burdens, unfortunately. And we mentioned also Erwin Neher several times. And for the people who do not know, he's a Nobel Prize he discovered together with, or actually developed together with Bert Sackmann, a patch clamp technology, which revolutionized neuroscience, actually, right? And I think this was worth mentioning. But really fascinating story, and I'm very, very happy that you shared with us this small insights, and it's quite intriguing and interesting to hear about it from first person. So now, when we know a little bit more about you, we can switch gears and go to science. What do you do in your lab?
Yeah, so we are interested to study and understand and then develop therapies for brain diseases. And for the large part, these are neurodegenerative diseases. So these are diseases where your nerve cells die eventually. And when the nerve cells die or are not functional anymore, then the brain is not functional anymore. And Alzheimer's disease, for example, leads to cognitive decline. This is what we know. We also have interest in other brain diseases, which are in part more neurodevelopmental. So we have a couple of projects and grants where we work together with colleagues in Munich, mainly on schizophrenia, on depression, on bipolar disorder. And we also are part of a couple of larger European Union projects where we look at, again, developmental diseases that lead, for example, to intellectual disability. But the connective aspect of all of this is actually that we are looking at epigenetic gene expression and epitranscriptomic at large, which we can discuss now what this in principle is. But I can give you an example for Alzheimer's disease. Why is it also so difficult to study this? So Alzheimer's disease leads to the loss of, all your abilities that make you human and your character. So cognition is declining, your social abilities are declining. And the big problem is actually that these phenotypes that you see, so the clinical phenotypes, once they occur, the disease is already incurable anymore because the pathology, so the changes that you see start 20 years before. It's almost like, in a way, some cancers you just don't detect and you only realize that there's something wrong a month before you would die or the patient would die. And that is the same with Alzheimer's disease. So we are very much focusing on methods for early detection, because this is probably really the key. So to find biomarkers that allow you to make predictions about the disease progression. and then also develop therapies that can be complemented with other therapeutic approaches and things like this. So this is what we study, and that's all arised from the original interest that I had to understand how is the brain encoding information and make it retrievable even years after you learn something.
This is, by the way, one of my interests that triggered me to go into neuroscience. So you mentioned several terms: epigenetics, biomarkers, even Alzheimer's. We are going to talk about them, but maybe we can introduce some general terms like what is DNA, what is RNA, and how is this related to the epigenetics, for example. And also, since you have said that Alzheimer leads to cognitive decline, and also many other neurodegenerative diseases lead to that. Maybe we can talk about one of the interesting cognitive functions of the brain, memory. So how do you define memory in your research? What do you understand under memory? And let's start with perhaps DNA, RNA, and how is this related to epigenetics?
Yeah, so the DNA is basically the building block of life and of human life, of course, also. And the DNA basically is encoding everything. So what I always tell the students is actually that you should think about this as a library. So we have about 200 different cell types, and every cell type, in principle, has the same genome. And the genome is encoded in this 4 base pairs AGTC of our DNA. And so that's the library, and now every cell needs certain books and chapters that it should read. So all the metabolic function, every cell has a Krebs cycle and does these jobs. But then there are certain books and chapters within these books that every cell has to read only if it's a neuron. So if you think about this, then actually every cell, so a neuron has the complete library, and it reads some parts of the library that any other cell also reads, but then there are specific parts of the library that only the neuron reads, okay? The same is for the liver cells. So there are certain parts of the library that the liver cell shares with other cells, but then there are some which only the liver cell needs. And now the big question is actually, if at the beginning of development, every cell has the same genome, how basically is this maintained throughout the life of the organism? And this is now when the term epigenetics, which means above the genome, comes into play. That's a term that was coined by Conrad Waddington, who was a development biologist there. And what he was trying to say is specifically this. He had this picture where the genome is a marble, and the marble can run down a valley or a hill. And there are different valleys, and it could end up in valley 1, 2, 3, or 4, and that were the different cell types. So in a way, epigenetics is everything that makes cells distinct and tells them which part of the genome they should use. Now, another example that might make this more understandable is, let's stay with the library analog. So if you have a library and it's full of books and chapters, how would a neuron now remember which chapters and which books it should read? And which has it read before, or if something happens, it should read this. So what you normally do, you put earmarks or bookmarks inside, right? And that's precisely what the epigenetic is doing. So let's say there's a learning event, and all of a sudden the neuron has to start some new gene expression programs. That means it takes a book off the shelf, reads a chapter, and now it remembers basically that this is something that I need, so it puts a bookmark in and puts it back. So these bookmarks are epigenetic modifications. And in reality, these epigenetic modifications are really placed on the DNA. So the simplest epigenetic modification is the methylation of DNA. So the cytosine can be methylated, and if it's methylated, normally that means that the gene is turned off, right? And it's a binary code almost. It's yes or no. The other key epigenetic modification that we have is the histones. So the DNA in every of our cell is about 2 meters long. So how does it fit into a nucleus, which is super tiny, right? So it's heavily coiled and it's wrapped around proteins, and these proteins are called histones. And now the idea is, if this DNA, 2 meter into 10 micrometer, is super condensed and wrapped, then it's not accessible. So the genes cannot be transcribed. Or let's stay with the library example, the books cannot be taken off the library because it's closed. Okay, and now there are mechanisms, and this is called epigenetics because, let's say, it's wrapped around these histones. So these histones themselves can be modified by all these modifications that we know: phosphorylation, acetylation, methylation, and so on and so forth. And there's a code that tells, for example, these genes, if, for example, histone 3 lysine 4 is methylated, then the chromatin opens up so the library is accessible and the gene can be read. If the same location now changes and it has histone 3, not lysine 4, but lysine 27 methylated, then the whole thing closes. And what people have done now is actually that they have tried to characterize, let's say, combinatorial patterns of these histone modifications that signal to the cell this part of the genome should be accessible and this one should not be. And this one should be closed forever. This one is closed but can be activated, and things like this. And what we know is that for various reasons in all of these diseases that I talked about, these epigenetic profiles change. That could have different reasons, basically. The enzymes do not work properly anymore, or there's just too much methyl donors, actually. So very simple approaches is to eat a methyl donor to have your epigenetic up to date. But there's also other evidence that this plays a key role in the diseases that we talked about, the brain diseases, because if you look at intellectual disability diseases and this whole neurodevelopmental diseases, where you have normally one mutation that causes then actually later on cognitive decline, then there are two groups of enzymes and proteins that are really overrepresented. The first one is, of course, everything that we know from this autism spectrum disorders, so really genes and proteins that act at the synapse. And the other one that's equally important is all these regulatory proteins that control chromatin. So I can give you a very specific example, if this is not too complicated. I told you that there's a particular pattern, so let's look at histone number 3. So there are different histones, right? 1, 2, 3, 4, and there are some other ones. And histone 3 at the position amino acid lysine 4 can be trimethylated. Then it's active. The gene is active. Now there are in the human genome and also in our nerve cells, there are 6 different enzymes that catalyze this methylation process. And there are a couple of more which also cause the demethylation. And it's always a fine balance that we know from phosphorylation. There are some enzymes that put the mark on the chromatin, and this we call writers. And there are some that remove the same mark. This we call erasers. And then there are protein complexes that read these patterns, and they are called the readers. Now, if you look at this histone 3 lysine 4, every enzyme in the human body that we know that orchestrates this process, when it's mutated, it's causing this neurodevelopmental intellectual disability disorders without any questions. So every single enzyme of the pathway, telling you that when you see now in sporadic diseases, so sporadic means we don't really know if there's a genetic cause, most likely there's one, but it's not one gene only. You also see the deregulation. So that's a super important process, right? And this is actually then something where people try to develop drugs that can reinstate this plasticity, this dynamic, this chromatin function, and then bring back the ability to learn or to recall information.
Mm-hmm. So you touched upon several interesting topics. So let's try to make the connection from memory to epigenetics and to this gene expression. We know that the biological unit, if I may say so, of memory is the synapse. We store information in the connection between two nerve cells. So something has to happen at the synapse, then something has to happen in the cell body. Is there any connection between these two units of the neuron, and how does it work in terms of memory, for example?
Well, of course, there's no conclusive answer because that's still an unsolved question. But of course, people can have hypotheses. And you mentioned basically that the memory is stored within the synaptic connection. I think also this is actually still a very valid hypothesis. But I think the ultimate proof for this is also not there, right? But it's very likely that the connectivity of the nerve cells plays a key role. And maybe I'd like to start with this. So Francis Crick, right, the person who described the structure of the DNA together with his colleague Watson, he became something like a neuroscientist in his later years, right? And he wrote another Nature article that I always like to cite, which is very interesting. And he was, in this article, which is just one page, addressing the question how, that we still have today. So how actually is an information maintained over years in a structure that is always turnover, like, and he was talking about the synapses then, right? And he says, so what is it then actually that is renewed, that is maintained, that keeps this information in this synaptic connections? And then in Germany, we have a saying, I say it now in German, actually, für den Hammer ist alles Nagel. So with other words, actually, for the hammer, everything is a nail. And what I'm trying to say is actually Francis Crick was, of course, very much interested in DNA. So it's not a big surprise that he came up with the idea that DNA is the key to memory. So there are various approaches. One is basically, of course, the only thing that does not change anymore in a post-mitotic neuron is the DNA. So the simplest view that came up was actually, okay, DNA methylation is a binary code. You have so many base pairs, actually, you could just store all the information there. But how does this come back to the synapse? So then he also said, well, there must be something at the synapse, which is probably maintained there. And of course, he said that's DNA, or that could also be RNA, he agreed upon. And he said there might be molecules that are self-replicating that maintain this, you know. And there is some evidence that we can discuss maybe that this exists. On the other hand, there have been other approaches. One of them is from Eric Kandel, so another Nobel laureate that got the Nobel Prize, not for something very specific, but more for his whole life work. And he, a few years ago, came up with some interesting data suggesting that there are prion-like proteins, basically, that are specifically at synapses, you know, that, you know, initiate folding and by this actually replicate a specific state or information that can survive this turnover of proteins because it's always renewed. But something like this most likely exists. And as Crick said, actually, so it could be DNA, it could also be RNA, and there have been some other interesting data on this topic. For example, this is also something that I use as a fun fact in the lectures. So there had been a very interesting psychiatrist in the 60s who did these crazy experiments with flatworms. And I don't know if you heard about this, but the idea was that you can train a flatworm to go to the left or to the right of a petri dish depending on the fact that there is some food reward or there is some danger signal. And what they did then is actually that they cut these worms into pieces, which you can do, and from every little piece there will be a new worm which would regrow because they are full of stem cells. And so first of all, they saw that every little piece of the worm could remember this information that they learned, but also they could feed these worms to other worms. And the paper even called actually transfer of memory via cannibalism. And then, so if the worm was eating the other worm that had learned something, they had the memory implicated. That's what they said. And then later on, they figured out that it's RNA. So they could just isolate RNA. And there has been even now some more modern follow-up data from more reliable scientists, basically, which show that this seems to be true. And the idea is actually that you can have RNA, which is modified, which you put then back into this nerve cell. And there it induces kind of epigenetic machineries that could dictate actually spines where they should grow, for example. Now, of course, you could say I'm also doing this hammer and nail thing because for me, epigenetics, everything is epigenetics, and that's where the memory goes. But there is actually some quite convincing evidence that maybe it's not the quality necessarily of the synaptic connection, but more the quantity to some extent, because there's evidence from data in model systems, but also from humans that we all know, that sometimes the memory might not be really gone, but it's just not accessible anymore. And the question is then actually, so you need to regrow synapses to make it accessible, but it doesn't seem to be so important where they are grown, but just that they are regrown. But to be fair, there's also evidence that actually the place sometimes matters, and people have ideas that it's the extracellular matrix, basically, which might still be maintained. So if you disrupt this, then they don't know where the connections should be made and things like this. So there are a lot of ideas. In essence, it's not quite clear what happens. I think the latest thing where there's a small but increasing research field is some people who try to find these engram cells. So the engram was coined by this German psychologist, and this is actually the nature, so the molecular basis of the memory that is formed. And the idea is there that we have now new labeling and imaging and optogenetic techniques that you can identify in model systems, of course, neurons that get activated when there is a stimulus of which we know it is learned. And then once you identify these cells and you can label them, you can ask yourself the question, is the same neuron that is activated during the learning also reactivated during the retrieval of the memory? And what happens then if I actually activate these neurons with optogenetic methods without actually initiating the retrieval? Would the animal show the same behavior? And that seems to be the case. Now, one fascinating aspect about this that I find is not only the fact that, in a way, it's very simple. So the nerve cells that become recruited into a circuitry would be the same ones that would be activated when you retrieve the memory. That's basically what this data says. But the interesting thing is that if you carefully look at the data, let's say there are 100 cells which got activated when there's a particular memory training procedure. Now, during the reactivation, yes, it's the same cells, but it's only 20% of them. So the other 80 actually don't get reactivated. And that might be very interesting backup mechanisms. So let's say the idea is always there's a stimulus and then there's a particular circuitry. So some cells come together and they make connections. And when this same set of connections is reactivated, then the memory trace can be retrieved. But obviously there's much more. And maybe this is the backup mechanism, that memories are stored in a larger network that you do not need. So in the end, you can lose information. You can lose nerve cells. And this is what we see also in cognitive decline. So the brain works fairly well, actually, as long as you don't lose, let's say, more than 30 or 40% of your brain. And this is also what we and others have shown in animal models. So let's say if you train an animal to learn something, and then you induce by genetic means neurodegeneration, and you do this, then let the brain degenerate up to 30%, you stop the process, and then you regrow the synaptic connections by some mechanisms. All of a sudden, the memory that was not accessible is accessible again, but that only works to a certain threshold. So if it's above 50% neuronal loss, then it doesn't work anymore. Mm-hmm. So this is maybe also why it takes so long, although the neurons are dying, until Alzheimer people really have clinical phenotypes, right? Mm-hmm, mm-hmm. So that is, I think, the big challenge. The brain is so plastic. And there are a lot of anecdotes. I think we all know, every other person has somebody in his family or in her family who is suffering from Alzheimer's disease, and then they are completely unable to remember anything, but then all of a sudden you bring them back for Christmas, home, and then they remember. So they have these bright moments, and that are not only moments, that are minutes and hours sometimes, and then they go back into dementia. And this clearly shows you that it's not always a loss of memory, but more a question of how accessible this thing really is. And the brain is super plastic. There's this other anecdote that I once read. So there was a person in the UK who was having headache, and they went to the doctor, they did an MRI, and they were shocked because there was no brain. So he had a super thin cortex, and in the middle was just CSF. And then it turned out that during birth, basically, there was a leakage of the cerebral spinal fluid. But because this was so early on, and the brain is extremely plastic, specifically during early adolescence, that that was pretty much compensated away. So it was pretty functional and led a normal life.
Mm-hmm. Yeah, I've seen this publication. I even showed it to one very famous scientist who is developing this theory for consciousness. Mm-hmm. And he was like, I saw this. I don't know what is this. Should I believe it, not believe it, how to explain it, etc., etc. Because this is a little bit off topic, but about consciousness, there are different theories about the interconnectivity of the system, etc. How complex it is.
Well, there are also some people, actually, it's always debatable if this is a disease, but there are a few people on this planet which cannot forget anything. So every piece of their life is there, which is a little, it's a handicap, right? Because you cannot have a normal life. Mm-hmm. But you can ask them actually what you did 20 years ago on this particular day, and they would be able to tell you. And of course, they are heavily investigated. They are still alive. And if you look at the structural changes and functional changes by imaging, there's nothing which is really dramatically different.
Maybe we lack the resolution to see exactly the...
Could be one thing.
Yeah.
I always like this article, I think it was also in the popular news right now about Einstein's brain, which was kind of stolen, and then they looked at it actually, and there's even the publication, I think, from the 80s, where they always claim Einstein had more astrocytes than other people, right? And that is still something that you often hear. But sometimes you have to do the fact check, right? So I read this paper, and it's let's say not a very optimal study design with an N of 1, which is Einstein, and then 11 other people, I think, which range from 30 to 90. And then they looked at 2 brain regions, and the data is not at all convincing. So I think there's no evidence that Einstein had more astrocytes, and that this is the reason. The hypothesis was that he has more astrocytes because the brain cells were better supported, right? Or he was thinking so much that he needed more astrocytes. Yeah. So.
Yeah, astrocytes are, by the way, another set of cells in the brain. So we have these neurons and glia, and astrocytes are part of the glia, which are believed to support the brain functions, and they're important in many other things, actually. Before we continue, I just want to elaborate optogenetics. This term that you already mentioned is a technique that can stimulate, so to say, cells that are genetically modified with the help of light. And also regarding this experiment with the planaria, which is fascinating, I recently watched another podcast where they discuss exactly this. And there are even bioarchive from 3 days ago showing RNA even that is not in the nucleus, but in cytosol, kind of make these epigenetical modifications in even several generations ahead in these worms. Exactly. And
so this is maybe something I can tell the listeners. We did a very interesting experiment, since you mentioned this, about this transgenerational stuff. And so there's always this idea, actually, if there would be mutation and selection, like the Darwin theory, that would sometimes be too slow. So why would evolution not develop something where you can, in a relatively short time period, respond to novel environmental challenges, but don't make it permanent, like a mutation and selection? And this is also what epigenetics in a way is. So a way to inherit acquired information. So in the most easy way, there's always this picture from Jean-Baptiste Lamarck, who died poor because people were making fun of him. He's not a real scientist. But he was against Darwin. So Darwin said, the giraffe has a long neck because there was mutation and selection, and the ones with the longer neck had then more children, all this stuff that we know. And Lamarck said, no, it's actually because they have a long neck. He never really said this, but this was his idea. They would have a long neck because they were stretching the neck. So, and this would be now inherited to the next generation. And of course, epigenetics is precisely this, more from a developmental point of view initially. So people were thinking, okay, so you are born and your liver cell always gives rise to the next liver cell, right? So the liver will stay the liver, and this is because the stuff that we discussed, there's the library, which is our genome, and the liver cell just know now, because of this epigenetic modifications, which genes they would need. But now there's more and more evidence from epidemiological data in humans and clearly in animal studies that there can be acquired information which is inherited to the next generation. And one super fascinating example was, this was a paper published by a group called Mann with the last name in 2010 in Nature Medicine. And they did not look at anything about learning. But, epigenetics is having a transient stimulus being transmitted into long-term adaptive change. So in a way, that's precisely what happens also with our memory in the cognitive field. But they looked at something which I found super fascinating. So they looked at liver cirrhosis. So it's a complex disease, right? And they took, in this case, an animal model. This was rats. And you can induce their liver cirrhosis also with a toxin. And then they had just 2 groups. So the one was the group which were injected with the toxin, and they got liver damage, and the other ones not. And then they were bred. So these were the fathers and the mothers, and they gave birth to the offspring. And again, the offspring, maybe they got the toxin not. So in the end, you had 2 extreme groups. And these groups were now challenged for the first time in their life with this toxin that would do liver damage. And the one extreme group was a group where the parents and the grandparents were not exposed to this toxin. So in other words, if you want to translate it to humans, the parents and the grandparents were not heavy drinkers. They had a healthy liver or were not eating fat and sugar. And the other extreme group were grandparents now, grandchildren, where the fathers and the mothers and the grandfathers and the grandmothers were exposed to this toxin. So they had heavy liver damage. Now the question was, if you put this toxin to these grandchildren, rats, what would happen? And the very fascinating observation was that the ones with the parents and grandfathers that had liver cirrhosis were protected. So there was something that was transmitted through the generations that protected the offspring from something bad that happened to the fathers and the mothers. So that's almost like in the Bible, right? Where you always, I know this by heart, Moses 20:20, we have to suffer for the deeds of our parents until the fourth generation. It's pretty precise, to be honest, actually. We know it goes until the third generation. Yeah, and then they showed that this is actually some super complicated process that involves DNA methylation, histone modification that obviously goes to the germ cells. So you do see this in the oocytes and in the sperm, which then through development would lead to a little bit more expression of a particular transcription factor that would dampen the response of the liver cells to this toxin. So clearly actually an environmental stimulus that is transmitted to the next generation. So I look at this actually as a shortcut of evolution where something really bad happens and now you have to protect your generations. But if the insulting stimulus is gone, then you also don't need this anymore. So it's not permanent, right? It doesn't mean that one mechanism, epigenetics, is better than Darwinian evolution. I think it just runs in parallel. And what we have done in particular is we are very much interested in preventive strategies. And the bottom line to this is that preventive strategies, so prevention is the best medicine, particularly though for Alzheimer's disease. And there are a lot of things, and that's the good news, that you can now do to lower your risk of Alzheimer's disease. And the latest data clearly suggests that 40% of the risk is modifiable by our lifestyle. And the two things that really stick out there is first, social interaction. So if you lead a socially stimulating life, which doesn't mean that you have 100 friends that are the best throughout your life, but you just have actually a lot of social interactions and talk to other people. And that actually, it's funny. So the data also shows that it doesn't really matter if you talk to somebody in real or actually via Zoom or something like this. So both is equally well. Not sure if this holds true on the long run. But this and the other big thing is exercise, aerobic exercise. And this is something that you can super easily study in animals. And we know this actually also from students. So if you do exercise, we can look into your brain and see that your hippocampus gets bigger because there are more connections. They did this even with students before they had to study for an exam, and the same thing. So exercising and cognitive training is improving cognitive function. And we talk about something like it's building up the so-called cognitive reserve. The cognitive reserve is everything that protects you from cognitive decline later in life. The mechanisms are not totally understood, but we know when humans and rodents perform exercise, they have more synapses, they are more maintained, the synapses are much more dynamic, and everything works better. Now the question was, is this also mediated via transgenerational mechanisms? And the short answer is yes, that has been shown by us and also by other groups. Meanwhile, that if you take, for example, animals, mice, and then the father or the mother or both run on a treadmill, they like this, and then they foster a child, then this child lives in a normal cage, but still has enhanced long-term potentiation. I'm pretty sure you will explain us now in a second what long-term potentiation is, because you can do this much better than I can do. And they also have enhanced learning and cognitive function. And this only goes to the one generation. And what we know now is actually that you can, of course, observe this as a phenomena, but in model systems we have the chance to manipulate this. And we identified two small non-coding RNAs. So these are regulatory RNA molecules that control protein function, gene expression, and all this stuff. So they're called microRNAs. And what we observed is that in the brain and in the germ cells, so let's say in sperm, for example, of these exercising animals, there are two microRNAs which are increased. They have the interesting names microRNA 212 and 132. And if you have more of these microRNAs, then the path of neurodevelopment later on from the fertilized oocyte is a little bit changed, that you have better connection of glutamatergic neurons, and the hippocampus is a bit better and a bit more plastic. And you could show causality, because if you block specifically these two microRNAs in the sperm of fathers that were doing exercise and do in vitro fertilization, you don't have this effect. If you put them in, you can have this effect at the same time. So very interesting, actually, that the evolution has mechanisms to manipulate what's in the germ cell, which would then slightly change the developmental path and make the organism different, better, for example. And there's another interesting aspect about this pathway, because these very two microRNAs are also genetically linked to schizophrenia. So let's say what people do in genome-wide association studies is they try to find variations in genes and link this to the disease. Because, of course, we have all the same genome, but we don't look the same. We are not all twins, right? So still our genome is different. And that is because the genes, certain positions, they differ. But we don't call this a mutation because it doesn't make you sick immediately. But you can see, for example, that people who have a certain variant of a particular gene do not get sick, but the risk is higher or lower. Now, people who have a specific version of these two microRNAs are more likely to get schizophrenia, which is precisely the other side from exercise. So brain development goes in the other direction, that it's actually not so plastic. Doesn't mean that you get schizophrenia, but you are more likely to. Yeah, it's the same. There are famous genes like BDNF, the famous brain-derived growth factor. So it comes in different flavors, and if you have one version of the BDNF, you're more likely, specifically if you live in a not so nice environment, to get certain psychiatric diseases. Mm-hmm. Yeah, but this is epigenetic in its core, if you want so acquired information that's inherited to the next generation. And of course, you can think further. So what would this mean for humans? That actually, so before you want to foster a child, you have to do a little bit of exercise and check your germ cells, actually, if these two microRNAs are increased, right? I'm not suggesting anything here.
Yeah, that's good that you made a distinction, germ cells, and we have somatic cells, right? They have different DNA, right? So it's interesting how the information translates when something happens in your brain, then goes into your germ cells, and this is maybe another field of research. I don't even know. LTP, so very briefly, this is, you can imagine it as a strengthening between the connection of two neurons that fire, so to say, at the same point, in a very, very brief terms. And also the hippocampus is an area in the brain which is important for memory formation and also recalling of memory. And it's very important also in Alzheimer's disease. And now you told us how we can protect ourselves eventually from getting Alzheimer's disease. But what causes it on first place?
Yeah. So what causes it is of course still a question of research. But there are two things which are clear. So we have in Alzheimer's disease the so-called familial cases, and we have the sporadic cases. And familial in this case means that there is a mutation in a particular gene, and this is the reason why people get Alzheimer's disease. And the very first patient that Alois Alzheimer was describing, this was a woman called Auguste Deter, who had Alzheimer's already with 51 years of age, which is super unusual. Normally, most people get it in their 80s, and if we would all live up to 120, most likely we all have Alzheimer's disease. What we know now is that Auguste Deter, who lived very close to here, so she was from Kassel and then was studied by Alzheimer, of course, in Munich. So she had a mutation in a particular gene called presenilin. And the presenilin is part of a complex that cleaves a protein that we call amyloid precursor protein. And this is another gene that, when it's mutated, we know can cause this familiar form of Alzheimer's disease, which is characterized by very early onset. So people get it in their 40s sometimes. And Alzheimer looked into the brain of Auguste Deter after she died, and he already found two things 100 years ago, more than 100 years ago now, that we still study today. And these were protein aggregates. And one of the protein aggregates that he discovered are the so-called amyloid plaques. What we know now is that these are aggregates outside of the cell, and they consist mainly of short fragments of this APP, the amyloid precursor protein, that is cleaved again by proteins which include this presenilin. So then this is one hallmark. So Alzheimer's disease patients have a brain full of plaques. And the strategy to treat them was to remove these plaques. There's one other hallmark that Alzheimer also already described in the brain of Auguste Deter. And these are protein aggregates that are now not outside of the cell, but inside the cell. And these are aggregates that mainly consist of a protein called tau. So tau is a protein that is linked to transport. It controls microtubuli. It's also found at the synapse. Doesn't seem to have a function that is so too well described. If you knock it out in a mouse, there's not much of a phenotype, for example, so it could also be compensated. But this is the hallmark of Alzheimer's disease and many other neurodegenerative diseases that collectively we call also aggregopathies, so some proteins kind of aggregate. And the idea is that, of course, if a protein aggregates inside the cell, in the end, every process will be disrupted, right? Because there are many things which are deregulated. It binds other proteins, it binds RNAs. And if it's outside the cell, of course, what you see in the vicinity of these plaques is that dendrites just die and spines and synaptic connections get lost. So this is what you see. This is a familiar case. And most of the model systems at the cellular level or the animal models are really based on these two models. So there are hundreds of animals, actually, models that have been generated which overexpress in various combinations mutant forms of this amyloid precursor protein or tau. Now, interestingly, tau is not mutated in Alzheimer's disease patients. It's mutated in another disease that we call frontal temporal lobe dementia, which also is close to Alzheimer, the second most often or the most often neurodegenerative diseases affecting people under 65 years of age, whereas Alzheimer's disease is then more for the older population. Now, the therapeutic strategies so far have really focused to remove the plaques and remove the tau. And for a very long time, this was not very successful. And there are many reasons for this. The first one is actually that, well, we do have these familiar cases, but we know also nowadays that one pill does not fit all. So it's a multifactorial disease with tons of disease factors. There's also not something like pure Alzheimer's disease. So specifically in older people, there's a comorbidity. So people always have a little bit of Parkinson, a little bit of that. Actually, some people develop dementia, which is the clinical outcome of Alzheimer, because they have heart problems and things like this. So probably there are very few people, which we call the sporadic ones, the older ones, that really have this pure amyloid pathology. Then also there's quite some data suggesting that there are people who have a brain full of these pathologies, but they had no Alzheimer's disease clinically, so they are cognitively totally intact. Now, how do we bring all this together nowadays? And there the point is that the clinical trials that we did for the last 20 years, they all failed. None was a success. In some of the cases, actually, even patients got worse when these kind of processes were manipulated. And there are two things to this. First of all, people were treated much too late. So in the early trials, I mean, sometimes there was no effect because the diagnosis was made, well, this is Alzheimer, by sometimes even primary care physicians. So God knows what these people really had. Very heterogeneous group of people that you treated. And then, of course, statistically, you don't see a difference, right? The other thing is actually that it has been ignored, that a lot of people actually have also other reasons in addition to amyloid that you would need to treat. So just get rid of the plaques doesn't make sense. Also, there was this theory that all these aggregates are actually something beneficial. So that, these little peptides are toxic, and there's a compensatory mechanism that puts us into these aggregates to put them away. And then if you release them, then actually it would get even worse. Nowadays, I think what we really clearly know is that we just treated the wrong patient population. Mm-hmm. Because, as I said, we know now very precisely that the pathology starts already 20 or 30 years before you see any clinical symptom. And that is important because the patient itself will also never realize this. So there's one very good criteria, there's even a scientific publication about this, that you don't have Alzheimer's disease if you come to the memory clinic and say you have Alzheimer's disease. Because normally it's the relatives that have to force the patient to go and get testing and things like this. But this is the point. And now there have been actually 3 trials that were kind of successful with the antibodies. So aducanumab was the first, lecanemab and donidalorsen is now there. And why are they now showing first signs of the fact that actually you can have a beneficial effect with an anti-amyloid therapy? Well, because the companies that run these trials now put a lot of effort and money into patient stratification. So now in these trials, which show first signs of being successful, actually you treated people who really are very, very early, and they confirmed by imaging approaches, by functional imaging, for example. You can look with a MRI scanner and see, do these people really have plaques and amyloid building up? And only these people who clearly have this and are very, very early in the pathology are now included and get this therapy. And then all of a sudden you see, yes, first of all, something that we always saw, the plaques are gone. So this always worked. So in a way, in the patients, you always saw what the animal model was showing you. So the animal models are all really good. But there was no improvement in cognitive function. And now, I mean, it's also not super dramatic and significant. It's debatable what it means if you increase by a few points on a scale. Because in the end, the question is, can the patient lead a life rather independently or not? And not if it increases 3 points on a cognitive score. But there are good signs, actually, that these things work. And that is because you not only remove this amyloid, but you do it really, really early. And that's what you need for a causative therapy. So if 50% of the cells are already dead, you can remove as much amyloid as you want. You will never see an effect. So the amyloid is gone, but there's no improvement in cognition. So this is why, in addition to the therapies that most likely do work as they should, as we know now, and they are already approved, we need to have ways to identify these people really, really early. Now the question is, how do you do this, right? And of course, you cannot do in practice then what they did in the clinical trials, so that you spend millions of euros to screen everybody above 50 with an MRI, PET scan, and CSF taken and all this stuff every other year. So this is why the field is now really moving to have markers that you can use at the population level for screening. And then you need liquid biopsies, but you don't want to have your cerebral spinal fluid. Actually, you want to have blood. And there is a lot of effort and also very promising data that at least what looks now, so when in your brain amyloid is building up, in the cerebral spinal fluid and in the blood, it goes down because it builds up in the brain. And this now you can very reliably measure in the blood, this drop of amyloid, for example. And that correlates well, has been shown with the MRI. So the idea is actually that you don't have to put everybody into a brain scanner, but you can first do a blood test, which would be, of course, much cheaper, much easier, and super fast to be done. And also that doesn't need much of an infrastructure, right? So this is one thing that people go about. And then there's great hope that actually when this will be available and you start with the therapies really early, that this would be super beneficial. Now, what is now on the market are these 3 different antibodies against amyloid, which seem to work very well. The question is this now something that you want to give on the long run? Because it's also super complicated and it comes with some really severe side effects. So if you give these drugs, probably every 2 months people have to monitor it by MRI so that they don't get any microhemorrhages or bleeding in their brain. So that's also not so easy. But there are ideas now that maybe you don't have to do this all the time. It's rather the strategy, if you identify people early, you give them, for example, an amyloid treatment with an antibody, but you give it just as long as the amyloid is cleared from the brain. And then you stop and then you give some other drugs, some maintenance therapies that, keep the status for a while. And then maybe in 5 years you have to do it again, something like this. And in addition, of course, you still need other therapeutic approaches that help to just increase the learning ability and plasticity. And this is something where we are working on now, in addition to finding biomarkers, to give, for example, epigenetic drugs, which are known to be key regulators of cognitive function. And if you inhibit certain molecules there at the epigenetic level, you can clearly see that there's a cognitive enhancement effect. So something like, a pill that would mimic the exercise effect that we just talked about, for example. So this is where the field is now going, and there are a million of other approaches. People also try to knock down tau with so-called antisense oligos and things like this. There are other therapies, anti-inflammatory therapies. I mean, everything that worked in animal models, but never worked in humans, for the very same reason that I just talked about with the amyloid. At the time point when the patient nowadays is diagnosed, it's too late. Because also epidemiologically, there's a clear correlation, people taking anti-inflammatory drugs for whatever reason, rheumatoid arthritis or something like this, they have a lower risk to get Alzheimer's disease, or cholesterol, actually, drugs that decrease cholesterol, for example. They also work, but of course not as a drug when you give them when the patient is already sick. So the future is actually markers for early detection and then combinational therapy. So probably we also go away from the fact that there's just one drug that has to be given. Most likely it's be a combination, maybe not all at the same time, but at different time points that will be combined. And for the early detection, I think there is this idea that there would be something like a dementia compass or something like this, where you have a set of 10 different tests, blood-based biomarkers, maybe some digital health apps, for example, where you can test your cognition, that people just randomly are offered to do once they are 50, and they can then screen their cognitive reserve, so to say, and then, when 3 of these 10 tests actually get down, then you get a message, okay, go to the memory clinic and have you checked up, and then they can decide if you have to get a brain scan or a more sophisticated blood test of amyloid, so that you have these people early, like they do in cancer screening programs, for example, which have been very successful.
Do you know how expensive are these treatment with these antibodies?
It goes about 20,000 euros, something like this.
Per pill, or?
No, that's not per pill. I think that's
procedure?
So 20 to 40,000 is per year. So that is expensive, but it's also not much more expensive than the antibodies for cancer treatment and things like this.
That's good to know. I did not know that we have this kind of a success. I heard about this drugs. I think even during one of your lectures 2 years ago, there was the trial with aducanumab.
Yes.
And I did not know that there is some kind of a success if you introduce the drug in the early stage.
Yeah, the latest drug now showed actually that all of them are very similar in clearing the amyloid out of the brain, but the latest antibody now showed even that there's an improvement in certain cognitive domains, about 30%. Or let's say not improvement, so it slows down the progression, right?
This is the thing, that we now try to slow down the progression. We cannot stop it. We don't know how to stop it. The only thing is to detect it early and try to slow it down. Right?
Exactly. And there are other preventive strategies. So the key would be really actually to build up your cognitive reserve while you can still do it, and then start any of the therapies early. And I think now, this is not the final cure now, but I think it's a very nice proof of concept. Because, there was always this huge debates also when I started in the field. So there was either tau, there was amyloid, and they were not liking each other very much, to be honest. And then if you were doing something different like epigenetics, people were laughing about you, right? What does this has to do now?
And now you go with the epigenetic markers that you try to identify to kind of screen or see or detect Alzheimer in the early stage. Can you tell us more about this research aspect of your work?
Yeah. So the bottom line is, as I discussed, that we need easy and screenable markers that are minimal invasive to detect patients early. And that does not mean that you want to detect really, from a diagnostic point of view, somebody who has Alzheimer's disease. But you want to detect people who have an increased risk. And of course, this you can do with genetics, but this you can also do with markers that you can read out, let's say, from the blood. And there are markers, proteins that you can measure, for example, light chain, NFL, that would increase, actually, if this shows up in the plasma, then that means there's some neuronal death, and things like this. And amyloid you can measure. Now, our approach is the following. So let's say, and I give you this example, inflammation is, of course, initially good. If it's chronic, it's bad. And that has also been a risk factor to Alzheimer's disease, and peripheral and central nervous system inflammation is there. And if you take drugs for a very long time, you have a lower risk. Now, how do you detect this now, for example, in any kind of liquid biopsy, let's say with the plasma? So let's say even interferon signaling. There are 100 different proteins that orchestrate this pathway. Now, our idea was that, let's say, neuroinflammation is a pathway, and to stay simple, there are 100 proteins that are relevant for this. And all stand for, let's say, interferon signaling. Now you go to the blood to a person and you measure 3 proteins, and they are increased, and that tells you, okay, so there's increased interferon signaling chronically, that's very bad. Now there's another person where out of these 100, there are another 5 proteins that go up, but not these 3. On the long run, it would be the very similar outcome, but now you have other markers that orchestrate this, because it's not quite clear where in the network now something happens, but the whole thing is deregulated, right? And the other thing is actually maybe, if you have 100 proteins which work in this pathway, then many of them maybe change just a little bit. That's not above the significant threshold, so to say. But now comes something interesting. So we have, in the human, we have about 20,000 genes that give rise to much more proteins because of splicing. So in the end, it's still a kind of complex system. Let's say you have 100,000 proteins or something like this that you could measure. Now, these proteins that act on a particular pathway, let's say inflammation, they are regulated again by, so to say, gatekeepers. And these are these small RNAs that we talked already about. They are called microRNAs. Now, microRNAs are little pieces of an RNA that can bind to a particular protein complex called dicer, and then they screen messenger RNAs, which messenger RNAs are the RNAs that are then translated into a protein. Okay? And now these microRNAs can bind this mRNA and lead to 2 things. Either the mRNA is degraded, but much more often it's just blocked, so it's not made into the protein. And the job of these microRNAs, and there the humans only have about 3,000, they are not more. And if you look to the brain, there are maybe 500 which are expressed. They regulate this pathway. So let's say you have 100 proteins and 50 of them and inflammation goes up. Then there's 1 microRNA that counteracts this pathway. So the important thing is actually you really deconvolute this whole pathway. Let's say I look at 100 proteins or I look at 1 microRNA. Meaning basically if there's a disease-associated shift in, let's say, the homeostasis of this pathway, then it might be much easier to look at 1 microRNA that tells you something about the whole pathway than to look at 100 proteins. So this was our naive point of view. And many people do this. So with other words, in a nutshell, what people do is they look at these microRNAs in the blood. Mm-hmm. Because there's another reason. They are extremely stable. Mm-hmm. So they are not degraded. And the other reason is actually it's known that they transmit information between cells. So these little microRNAs can be packed into little vesicles that are released from the cell. And that could be a neuron in the brain. And then they not only go to other neurons or other neuronal cells, they can also be released into the circulation. So you have them ending up in blood. Mm. And that's known. So there are, for example, brain-derived microRNAs or other RNAs that you can detect in plasma. So the idea is that you have a window, like similar with the A-beta. So you have a window to the brain by looking into the blood. And that's what we do. So we sequenced these microRNAs in large cohorts of Alzheimer's disease patients, or more importantly, patients that do not have Alzheimer's disease. But we know, because we longitudinally followed them up, we know that 2 years later, many of them got Alzheimer's disease. And then you can ask the question, is there anything that I see in the blood that changes that would allow me to predict which of these people will have Alzheimer's in 2 years? And there we indeed found some signatures. For example, we found 3 microRNAs that if you measure them together, one stands for synaptic plasticity, one for inflammation, and one for basically metabolic processes, then we know that we can make a prediction. And what we do now, of course, we don't want to sequence these things. We want to have an easy assay, and we are developing together with a company that's called Fasisi here in Göttingen, or we are trying to develop an assay that you all know, that's this corona lateral flow assay. Mm-hmm. So, but we don't detect the virus because we adapted the system that you can also detect an RNA. In the end, it's a pretty easy chemistry because on this lateral flow, you can detect just anything. So in the future, it would be interesting if you just take a drop of your blood, put this on this test, and then you can see how much of this microRNA you have. And if this is increased, then you can go to the doctor and say, okay, I want to have also my A-beta measured in the plasma. If this is increased, then you can say, okay, so now you either have to change your lifestyle, you can do this and this and this, and we also monitor you closely with an MRI. And then if the amyloid really builds up, then would be the time to get, for example, this antibody. And then hopefully the idea in the future would be that these people don't get it then actually for the rest of their life, but you clear out the A-beta once, and then you have some other maintenance therapies, and in 5 years you come back and see, do I need to do this again?
Do you know the timing from discovering this mRNA and then the onset of the disease, for example?
Yeah, so what we now do is that we look into the healthy population, which is, of course, always a bit complicated. What we know from the studies that have been done is that once it's really increasing, you have about 2 years, which there has to be something else, right? So because we are talking about the 20 years, so with this signature, we still capture the individuals that are already advanced. It's better than what we have before, but they are already advanced. And to do these other studies is, of course, a little bit more challenging, but that's what has to be done now. So in principle, the ideal study would be that you take people from their 20s and follow them up for the rest of their life and do a deep phenotyping every 2 years. Now, fortunately, actually, some people did already such experiments. We are collaborating, for instance, with something that's called the Framingham Heart Study. So this is also in the Boston area. This is a longitudinal study that runs from the 1940s, now in the 5th generation or something like this. And they have they started, of course, for cardiovascular questions, but they have people now where you have very pure Alzheimer's disease, you have the post-mortem brain of them, and you have blood samples, let's say dating 30 years back before they was diagnosed with Alzheimer's disease. And of course, these samples are very precious, but due to some colleagues that we also still have from Boston, we are now in an NIH grant together and are allowed to analyze these samples. So that would be super interesting, of course, to see what will happen there.
That is fascinating. Yeah. Interesting work, and I'm looking forward to find out more about it in the next months, years. I don't know how long it will take. Thank you very much for summarizing your research and also telling us more about Alzheimer, the things that you're doing, and also in general about the stories that you shared with us. I would like to switch the topics since we are progressing in time and we decided to keep it shortly before this podcast. And I would like to talk to you about some problems in science. Right? We everything that we discuss is fascinating, is very nice, and we enjoy doing science, I believe. But as any other field, there are problems in our field as well. Some of them we have already discussed on our podcast: reproducibility of experiments. People who are trained, for example, postdocs, they leave academia, they go to the industry. We have also difficulties in the publishing or getting grants. There are many, many domains that we can improve our field. If I may ask you to pick one, two, or several and say, why is this for you a problem and how could you overcome it as an advice for us, for the younger generation, for example?
Yeah, I think the points that you mentioned, they are always, I would not call this problems necessarily, but challenges. So I think we are living, as scientists, I think the biggest issue is always to get, the funding to do the science that you want. And of course, if the economy is not going well, or there are other gross political issues that affect the whole world, then the first thing you can do is to cut science, right? And I recall we always used to say Alzheimer's is a devastating disease and the numbers will increase, actually will double by the year 2050. That is not something actually I learned, and also we were discussing that you should say, because politicians, of course, don't think in the next 30 years, but they think actually, if you're lucky, actually by the next 4 years. So telling them that Alzheimer's is going to be a big problem in 2050 actually is not really raising the awareness. But of course, then you just have to say, well, it will be an increasing problem, but it is a problem already right now. And there you just always have to fight for this. And I think you need to have the stakeholders understand that it has to be a mixture of, let's say, blue sky research. So you just have to do something that may not be applicable immediately because you never know what comes out. And at the other hand, I think it's not bad that you have these structured programs like these health centers, of which the DZNE is one of them, where you say, okay, so our topic is we have to work on neurodegeneration and things like this, right? And the other thing is actually you have to tell the public, I think, why is this important in layman terms, so something that you do here with your podcast right now, right? So this is one issue. The other issue is, which you mentioned, which I think is very important. So how is the career planning? I think the fact that the universities or also the extra university institutions educate students and then they go to industry, that's our job. So this I wouldn't see as a problem. I think that's what the universities are there for. And if they go and do good science and do good research in industry, perfect. If they stay in academia, that is also good. I think an issue that you mentioned, but that also goes back to issue one, which is simply the funding, is, of course, how do you have sustainability, basically, for people who want to do research, but they don't want to rise to the professorial ranks or things like this, right? So what we called, and I don't know how to translate this in English, maybe because it doesn't exist there, is this akademischer Mittelbau. So, people who have a permanent position and then they do things. And this is actually something really, really important. Because as you mentioned, you train people, you have them educated, maybe they stay as a postdoc and then they leave and then the whole technology is gone. And this is actually especially difficult in the area that we live right now where everything is AI and you do bioinformatics and you have to do this and here and that. And of course, to get people trained that they do understand the question, the biological or medical question behind this, and at the same time, have all the computer skills actually is, I think, what we need, right? And then you have to foster them. And there's no good solution to this, of course, because I think the initiatives to say, well, you can only work for a certain time period in academia and then you have to make permanent is, of course, a nice idea that is completely unrealistic because we would love to do this, but you cannot do it because then you need board funding. Board funding means that you have the position. Let's say if I hire a person who's 30, then you have to document that you can pay this person for the next 35 years. And that you can only do if you have guaranteed funding that is not third-party funding, right? And on third-party funding, you cannot do this. So you can say, I'm 100% sure that I will always have a grant to pay you, but then the contract cannot be issued because it will be illegal, right? So the whole system does not really work, right? And the solution to this is very easy. I mean, give the universities and the institutes a decent amount of core funds. Then, of course, they can also have these people, actually, they can get permanent positions. Now, this is unlikely to happen. So this is why we have to find this compromise. And, I mean, all these ideas that they also put forward, they will not be put into reality, right? So, but that is and I think, in my view, this whole idea with the öffentliche Dienst is a little bit bizarre. So this is different in the US, right? So in the US, again, I mean, this is how we started. Maybe that's a good end. So there, I mean, you can, in any position, of course, rise much higher, can also personally make much, much more money. But there's also not the same level of security here. So here, we always want to have the security. We have a permanent contract. A permanent contract at the university means actually that this is it, right? So you can go to work, you cannot go to work, doesn't really matter. That's a bit extreme, right? But then that's it. So even öffentliche Dienst, that was it. And in the US, actually, no matter at what position you are, they can say, sorry, that's it. So take your stuff and you're out, right?
Okay. So the tenure tracks are...
Well, not at the tenure track, but actually even at the industry and all this stuff.
Okay, yeah.
So they could, and there actually, I mean, if you're not at the state university, what I don't know if you know this, but a significant amount of your person's salary has to come from your grants. If you don't have a grant, that is very likely that actually for half a year you don't have any income anymore. So,
but can you get more than...
That is the other thing. If you get more, of course, you can also increase. This is what I mean. So you don't have the same level of security.
If I may stop here, is this incentive to do bad science, for example?
This I don't know, to be honest. I mean, that's always what is debated, of course. Also because if you don't have a grant, then I mean, you might be under personal challenges, right? If you have a mortgage for your house or things like this, right? I wouldn't go so far, basically. I think also this whole field of, you know, what they say in industry, that academia science is not reproducible and all this stuff. Well, you can also go the other way around, because I've been at quite some companies, and actually, I mean, the research that they do is not so much different from ours. I mean, they don't necessarily have the pressure to publish, but they have other pressures and other deadlines. And I honestly think that sometimes it's not so much that something is, you know, willingly actually manipulated. I mean, that of course happens, you know, but very often it's also other things. So, you know, the model systems, the things changes and all this stuff. And what I found super fascinating is, and there we go back to the epigenetics. So if we have this transgenerational effects, right, that there is a challenge from the outside, and then something happens from the brain, for example, via this microvesicle, something goes to the germline, and then, you know, this is trying to protect the next generation from something bad. Very often, actually, in the field, it was, somebody made a transgenic mouse, and then, the phenotype is not there, it's not there anymore, right? But now think about this, and we looked into this, it's not yet published. Let's say you have a mouse that produces a lot of amyloid plaques, right? So if this whole thing would be true, then there should be some mechanisms at place that protects the next generation from the amyloid plaques. And that's sometimes that you see. So the phenotype kind of washes out. There's, of course, genetic reasons for this, right? But there might also be, on the short run, this epigenetic reasons. And indeed, when we compared, for example, APP animals that are always, you know, now it becomes complicated, but heterogeneously bred. And then you look at wild-type animals later on. There will be always wild types, totally healthy animals. But let's say they had a transgenic heterozygous father or a normal wild-type father. They're hugely different, their transcriptome and their sperm cells. So that means these mechanisms are at play. And then all of a sudden you have a lot of animals that show the phenotype, don't show the phenotype, and things like this, right? So these are things that you never thought about, right?
This is very interesting, because could it be now that we invest 20 years and billions of developing drugs that will not work in our generation, for example?
Well, the good thing is actually that whatever we know about this epigenetics is kind of short term, right?
Yeah.
So, I mean, in our case, what we have seen really is only that it is one generation. Some people have seen 3 generations, but normally even if they went longer, so if the insult is not there anymore, then it kind of washes out. I think that's, if we trust that this exists, that's part of the mechanism, most likely. But what I'm trying to say is actually that, and we know this from the journal club, so when you discuss, this data is complete bullshit, and I don't believe anything, right? I think that's of course almost like a reflex, actually, when there's something published in a big journal that cannot be true. And there's always, you know, people say there's this correlation between high impact papers and not reproducibility. I think sometimes the models are just too complicated. I mean, look at everybody works with iPSCs, right? I mean, there's a huge heterogeneity between these things, and you have the different clones and all of this stuff. I think, in a way, what I think should be, but that's a question of money, and what could help this is actually just to increase the N number dramatically. So honestly, we all do this because you don't have the funds and all this stuff, and then you do an N of 5, some people do an N of 2. And then I really say also when I review the papers, so they do a gene expression with an N of 2. I mean, that could be, you never know if this is now by chance. So this actually, if you really do a seriously power calculation, we all have to work with an N of 50 or something like this, right? Which we don't do. I think that is more of an issue.
This is more money, more time, and more people involved, I guess, more work. It's hard, It's doable maybe, but for some other models, for example, N50 with monkeys will never happen.
No, of course not.
N50 with flies is a daily
It's
routine.
Also not every time that you need this. You know, it's also if you reproduce it at least once or twice, I think, then it's okay.
So take-home message for the young scientist: get more money and do better science.
No, the take-home message is actually that once you do your PhD or your postdoc, this is if you want to continue in research, no matter if this is now academia or if this would be at the industry, or basically if you want to go into some science administration. I mean, this is the time where I think you should decide if you really want to do this. And if you want to do this, then that's the time where you decide the rest of your career. So during this time, and that is something that sounds a bit stupid, but this is what I've seen throughout my career when I was still there, and the people, there is definitely a correlation between the people that worked super hard during this time and had more work than life balance during this time only, and then made it and became successful, right, than the others. So this would be the time, I think, where you should really invest. But that's not for everybody. So I think there are many other jobs. But then it becomes, of course, harder because there is no akademischer Mittelbau anymore, right?
Yeah, but this is some other aspect that not everybody can be a PhD student also, right? I mean, at least I think so. But this is completely other topic. I think it's a nice point to stop right now here, our conversation. And maybe next time we can invite you when you have more information about the ongoing research and share with us your recent papers.
Happy to do that.
I would be really, really keen on finding more about it. Once again, Professor Fischer, thank you for coming. It was a pleasure talking to you and hope to see you again around.
All right. Thanks a lot.
Neuroscience and beyond. Know more. Get inspired.